process that selects for preferable traits. So here's an example. Darwin, he found these finches on the Galapagos Island, and some randomly had developed a better beak for eating seeds. The individuals that had this better beak survived better, and they ate more, and they reproduced faster. Since they could reproduce faster, they were able to spread their DNA through the population more efficiently to more organisms, and then their offspring were able, since their offspring were in higher proportions, their DNA got spread through the whole population. Is that clear? Does that make sense? Did I ramble? Good? Okay. Huh?
Not enough for
That's macroevolution. It's evolution over a long time in large organisms. I did microevolution. That's stuff that's really small. This is 2 microns, the size of this stuff. Microevolution, the nice thing about microevolution is that it happens really fast. This E. coli reproduces every 20 minutes. It splits into 2 cells. So for scientists, it works out really nicely to use this stuff because it can reproduce fast. You can run experiments with it and look at 20,000 generations in a year. So here's the basic outline for it. We took our original source of E. coli, split it into 4 lineages. 2 were put into the higher temperature environments, these 2. And the other 2 were put into the lower environment temperatures. We kept on, every morning we put them into a new solution. This is called laurea broth, and it's like a nutrient broth that has all the required nutrients for the E. coli to grow. By the end of the day, in 24 hours, they would run out of nutrients and we had to transfer them to a new batch so they wouldn't die off. That's what each section was. So we had like 3 sections here, for example. Fairly simple. We did this for, I think, 2 months for running, and we're going to plan to run it all the way until June to see our results. And then at the end, we're going to compare their reproductive rates. This is the actual layout. It's a little bit more complicated, but basically what I saw on the previous slide is the green section. We repeated that every day, transferring 50 microliters from the old batch to the new batch. At the end of, in the end of June, we're going to send off samples from each 4 lineages to a lab and get DNA sequence to see if there's actually any differences in it. And then we're also going to do our own comparative analysis of it. We're going to mark our colder lineages with a genetic marker. That genetic marker is going to turn it so you can see the difference between this hat and that hat. It's going to turn these green. It's going to leave these fine. So then we're going to take the ones that do take up the genetic marker and put them into a new solution. That's this layer here. Then in this layer here, we're going to have, since they're all going to be growing at different rates because of the temperature difference, we're not going to have the same density in all 4 lineages. So we have to use photospectrometry to find the density of each 4 lineages. Using that number, we'll be able to standardize the concentrations by adding different amounts of water. So at this point, here, all 4 will be at the same concentration. We'll take those 4 and then mix them into comparative plates. So H1 will go here, and then we'll combine this one with here. You can see all the lines. And these are plates here, they're not these vials here. So we'll be able to actually see the different colonies. We'll be able to see that there's the clear colored ones and then there's the green colored ones. We'll be able to count, and then we'll be able to compare which one's reproducing fastest. And we're going to do this twice, once at 44 degrees and once at 34 degrees. Hopefully, the ones in red, the hot temperature, will reproduce faster and better at the 44 degrees than the cold ones, and vice versa for the other ones. That's the extent, and that's natural selection, basically. So the question is, how does this relate to modernity? Well, it's related directly to Darwin. Darwin came up with all these theories. He was the pioneer in all this. He came up with evolution. He came up with natural selection. And he was really a perfect example of modernity because he completely was innovative. He completely reformed it. It was all new. And another great thing about Darwin is that it's all still true. It's all still modern also. It's all still current, and those theories are still used today. So that's the end. came up with this idea, and then I found out